Okay, so thank you all so much for coming. Whether you're here in the Portland office or elsewhere, potentially in space-time, this talk is an invertible transform for efficient string matching and label-directed graphs. And before I explain why this is interesting, I'll tell you a little more about myself. Okay, so my name is Abhinav Nellore, and I'm right now faculty at Oregon Health and Science University, OHSU. My PhD is in theoretical physics. I worked on using string theory to help understand phenomena like high temperature superconductivity and the equation of state of matter formed in particular particle colliders. But for the past decade or so, I've worked on problems in and around bioinformatics. Now, a lot of this work has focused on improving the queryability and accessibility of archived publicly available genomics data through their uniform reanalysis and through the release of summary outputs. This has often involved secure data analysis in the cloud with, for example, Amazon Elastic MapReduce, but not always. And among the not always is an effort I'd like to highlight called recount methylation, whose development was led by my former PhD student, Sean Maiden. So DNA methylation is the most widely studied epigenetic mark. It can, for example, silence genes without changing any of the underlying DNA sequence. And recount methylation is an R bioconductor package and accompanying database spanning over 100,000 human DNA methylation samples. At OHSU, my group has also worked on computational problems in immunology and cancer immunotherapy. Together with Mary Wood, Reed Thompson, and others, we developed NeoEpiscope, software for identifying cancer-specific proteins called neoantigens from tumor DNA sequencing data. Neoantigens can potentially be targeted by vaccines to engender or enhance an immune response to cancer. At the outset of the pandemic, we kind of shifted gears and attempted to predict associations between immunogenetic variants and susceptibility to SARS-CoV-2 as well as severity of disease. This was one of the first analyses of its kind. It's an effort led by another former PhD student, Austin Wynn. But today I'll talk to you about nation efforts by my collaborators and me to develop tools for graphs encoding genome variation. So I'll begin this talk with sort of a crash course in the relevant and motivating genomics. Why do people care about graphs in genomics, and why might Galwegians, okay, why might Galwegians care as well? Then I'll describe a method I developed with Austin Wynn and Reed Thompson to index label-directed graphs for efficient string matching. Now, this method relies on a particular random-like sequence, and I'll close out with work I did with Rachel Ward on defining the sequence properly and constructing one efficiently. So let's dive in. Now, a genome is all the DNA in an organism. And genomics is a study of genomes, their structure, their function, how they evolve, what happens when you edit them, and more. Human genome spans roughly 3 billion base pairs across 23 chromosomes, which themselves generally occur in pairs, one from each parent. And between any two individuals, roughly 99.5% of that sequence is identical. At its lowest order, the structure of DNA is a double helix that doubly encodes a sequence on an alphabet of 4 nucleotides containing the nitrogenous bases A, T, C, G. And the central dogma of molecular biology is often stated as DNA is transcribed into RNA, which is then translated into protein. Now, while that particular statement misses some non-trivial details, it does capture the essence of information flow in molecular biology, how variation at the genome level gets propagated all the way up to phenotype, observable differences among individuals. So 99.5% identical between any two individuals. What's in that 0.5% that's different? Well, there are short insertions and deletions of sequence in one genome with respect to another. And there are single nucleotide variants, differences of a single base in a stretch of otherwise identical sequence. But there's also more involved variation. Structural variation is defined as divergence of more than 50 bases in one genome with respect to a reference genome. And that includes long deletions of sequence, long insertions of sequence, includes mobile genetic elements such as transposons, which implement a cut and paste mechanism in genome, and retrotransposons implementing a copy paste mechanism in genomes. Then there are tandem duplications, which means that it's duplicated sequence that occurs side by side in a genome, and interspersed duplications, when it doesn't occur side by side, it's distributed. Finally, there are inversions and translocations, which occur when a piece of a chromosome breaks itself off, and reattaches itself in a different way to either the same chromosome or a different chromosome. A class of variants I'll keep coming back to are tandem repeats. Now, they are grouped into mini satellites and microsatellites according to what the repeat unit is. Tandem, again, means side by side. So mini satellites, the repeating unit has 10 to 100 bases, and for microsatellites, it's less than 10 bases. Microsatellites are otherwise known as short tandem repeats, or STRs. And on this slide, I depict an example of a short tandem repeat. The thing that differs from individual to individual is the number of repeats, the repeat count at the STR locus. So there's all this variation among humans, and how do we know what's different? Well, we've sequenced their DNA, and while I'm not going to go over how DNA sequencing technologies work, that would be a whole other talk. There are a few key facts I want you to keep in mind while I progress. And number one is we never really get from any of these sequencing technologies available today the full extent of a chromosome from end to end right off the bat. Instead, what we get are DNA sequencing reads, literally readouts of segments of DNA sequence that are generally ultimately obtained from randomly fragmenting genomes. Number two, there are 3 generations of DNA sequencing technologies, and they differ more or less, well, 2 of the ways they differ are in the lengths of the sequencing reads they output and in their base calling accuracies. Now, first generation of DNA sequencing technology is Sanger sequencing, and it gives reads that are between 500 and 1,000 bases long. It's a lower throughput technology, but it's highly accurate. Its base calling accuracy is 99.99%. Then in the 2000s, second generation sequencing became commercially available, and it allowed us to really scale up the sequencing we could perform. It's otherwise known as next generation sequencing or massively parallel sequencing. Now, it gives us reads that are shorter than Sanger sequencing, between 50 and 500 bases long. Its base calling accuracy is also up there. It's above 99%, not quite as high as Sanger sequencing. Market leader in this space is Illumina. You may have heard of this company, Illumina, if you're, I don't know, interested in companies. And now there's there's also now 3rd generation sequencing, which you'll hear about more and more. And the biggest players in this space are Pacific Biosciences, or PacBio, and Oxford Nanopore. So PacBio's technology gives reads that are tens of thousands of bases long, and Oxford Nanopore hundreds of thousands, even millions of bases long. I think the record is 4.3 megabases. Now, these technologies are amazing. They've also historically lagged first and second generation sequencing in terms of accuracy of base calling, but the manufacturers have been pushing that accuracy up to the upper 90s. And these technologies are really coming into their own, they're really maturing. So it's an exciting space to work in right now. Now, we're not getting chromosomes from end to end from DNA sequencers directly. So we have to assemble these chromosomes. It's a process called genome assembly. And what we're doing is we're taking these sequencing reads, we're stitching them together. So they have overlapping sequence. So you can imagine stacking them one on top of the other, on top of the other, on top of the other. And then obtaining long contigs, regions of contiguous sequence, which hopefully ultimately can stretch from one end of a chromosome to another. But, this assembly process doesn't really work as well when there are regions of the genome that are repetitive. It doesn't work as well with repetitive genome sequence or with very short reads, because both of these give rise to less unique sequence in the genome, which means that you can't necessarily place them confidently. What that does is increase the space of possible configurations in which you can assemble a genome when there should be more or less one correct way of assembling a genome. So in terms of, as far as human genome assemblies are concerned, in 2000, the Human Genome Project announced the first draft of the human genome. And it covered roughly 92% of that genome and included euchromatic sequence alone. Euchromatin is lightly packed DNA. It's in contrast to heterochromatin, which is tightly packed DNA, more difficult to access, contains more repetitive sequence, and it sort of eluded sequencing technologies of its time. In 2004, that same consortium released a finishing euchromatic sequence, and shortly after kind of passed the torch to the Genome Reference Consortium, which now regularly releases, semi-regularly releases, mosaic assemblies of the human genome and patches that correct regions that have been gotten wrong. Now, what's interesting about these assemblies is they are mosaics, they are from multiple individuals. Those individuals are anonymous volunteers from Buffalo, New York. Just kind of random. It could have been anywhere, it could have been Bombay or Bamako, but it was Buffalo. For these recent assemblies, roughly 70% of the DNA sequences from a single individual who's likely African-American. What makes this really interesting is that bioinformaticians around the world generally use these assemblies released by the Genome Reference Consortium with respect to which they measure variation in other genomes they have. So we're looking at variation from the perspective of this reference, which is dominated by a single individual. And that means that these analyses can occur biases if you're working with genomes that are more distant from that individual. And you might be able to infer the effects of this later in the talk. Now, last year was a seminal year in the world of genome assembly, because it was the year the telomere to telomere consortium, or T2T, announced the complete sequence of a human genome. One genome, including both euchromatic and heterochromatic sequence. It's a very impressive achievement. You can argue that we didn't actually sequence a human genome until very recently, and was made possible by third generation sequencing technologies. They use both PacBio and Oxford Nanopore reads to be able to pull this off. But things aren't just exciting in the third generation sequencing world, because it's the second generation sequencing world. These short reads that the market of which is dominated by Illumina, that are driving down the cost of sequencing and have been since the mid-2000s. So you can see that sequencing costs have dropped dramatically since the mid-2000s, faster than the expectation from Moore's law, which is that you'd have the cost of sequencing every couple of years. So much so that there's a startup, Ultimate Genomics, that last year announced the $100 genome. I don't think that their technology is right now commercially available. Perhaps feeling some pressure, market leader Illumina announced a $200 genome a few months later. I'm sure that makes sense. At the same time, genomic data on public archives has exploded. Okay, there are now over 17 petabytes of data on the largest of these archives, the Sequence Read Archive. And investigators are required to deposit their data on these archives by federal funding agencies when they're publishing their papers. So it's composed of both controlled access and public access data. The controlled access data is human. And if you visit the Sequence Read Archive website and search for human DNA sequencing data, you'll find that roughly 95% of samples are Illumina short reads, okay? So if short reads are most the reads on these public archives, and short reads are bad for genome assembly, how are you supposed to analyze them? The answer is alignment. We align these reads to pre-existing genome assemblies, like those released by the Genome Reference Consortium. The way this works in software like Bowtie 2 is that we build a suffix array of that genome assembly, could be a compressed suffix array. Then alignment proceeds in two stages. There's a coarse grain stage where we sample short segments of sequence, seeds from a read, and we exactly map those to the genome assisted by that suffix array, and this takes time linear in the size of the seeds. Then we pursue a fine-grained alignment step where we use a dynamic programming algorithm like Smith-Waterman alignment to infer variance in the read with respect to this reference genome. And this takes time, O of nm, where n is the length of some sequence that you pull down from the reference. So what your seeds have done, I neglected to explain this, is give you a consensus of where the read should align, and then you pull down the shorter sequence from the genome, and then you perform this, you do this Smith-Waterman alignment step, you find where the variants in the read are with respect to the reference, and it takes O of nm time, where n is the length of the sequence you pull down, and m is the length of the read. So alignment to a single linear reference works great for detecting short insertions and deletions and sparse single nucleotide variants, but it doesn't work so great for detecting structural variation. You can, for example, imagine a read that is entirely contained in a very long insertion. That read then doesn't have any anchoring sequence that's contained in this reference genome assembly. So there's no way for us to place it in the genome. And that read remains unaligned. Also doesn't work so great for dense single nucleotide variants. In this first coarse-grained alignment stage, you'll have these seeds that are just bouncing off the genome. We can't actually locate them because there are no exact matches. Doesn't work so great for tandem repeats either, because these repeat counts are highly variable. There can be a lot of them or there can be a few of them. So looking again at that short tandem repeat example, what if instead of representing a genome as this sort of single linear reference text, we instead used a graph and represent that short tandem repeat as a cycle in the graph? And if we take a read and we were to align it to the graph, it might align across A, T, A. Oh, good, you can see my pointer. And then T, T, A, T, T, A, an arbitrary number of times, could be a million, and then come out G, A, C. Okay. We could also imagine like representing all sorts of variants using graphs as alternate walks, SNVs, deletions, insertions, inversions, duplications, and where alignment of reads to a single linear reference may give us a lot of unaligned reads, ones for which we are not able to obtain a match, thereby aren't able to detect variation that may be in the sample and recognize it in downstream analyses. We can align those reads to the graph across these different classes of variants. If they're present, then we do detect the variation. We can actually recognize that variation in downstream analyses. So turns out that there are lots of folks in genomics interested in this way of doing genomic analyses. And there's a human pangenome project that was announced just last year as a resource to map genomic diversity. So what they're doing is using the same techniques that the T2T, the telomere to telomere consortium used to sequence a single genome, sequencing many genomes, and then building a graph representation of that variation. Just last month, they released a draft human pangenome reference spanning 47 genomes. And this is a draft. There's still a lot of debate about how to properly how to properly structure variants in the graph, whether or not certain paths should be merged between chromosomes, the extent to which it should reflect evolutionary history, et cetera. And just last week, a Chinese pangenome consortium was announced. There's this paper, Gao et al. In Nature, and they released a pangenome graph covering many genomes of 36 Chinese populations. So this space is heating up, but graphs representing genome variation isn't the only reason to care about graphs and genomes. They can also serve as a tool for protecting genomic data privacy, which may be of interest to folks in the Galois community. So why do you care about genomic data privacy? Well, while the Genetic Information Non-Discrimination Act of 2008 offers theoretical protection from discrimination on the basis of genetics in employment and health insurance, it doesn't cover all forms of insurance. And further, legislation like this doesn't necessarily offer immediate protection. Things could get litigious and arduous and drawn out to prove that there was such discrimination. Now, two, a malicious actor who has access to your genomic data can broadcast health information inferred from your variants, like disorders. They can discover or broadcast your biological relatives, which may be undesirable for some individuals. But then there are possibilities that may initially seem outlandish, but may be realized in the not too distant future. You can imagine a malicious actor building a bioweapon that targets specific variants in your genome. Maybe a company could make a product from your DNA without your permission. You can imagine, say, a company advertising, making your baby look like a celebrity and not getting the celebrity's permission to access their DNA. So this is all very murky territory. So it seems prudent to practice a principle of least privilege when it comes to genomic data access. I just want to emphasize that these variants in your genomes can be highly identifying. And in the United States, it's standard now for forensic scientists to use the repeat counts at 20 STR loci to identify individuals from DNA collected at crime scenes. And individual structural variants can be rare, so much so that in large cohorts of hundreds of thousands of people, you hear people talking about private variants that are present only in single lineages in that large cohort. So how can graphs help? Well, in that STR example, when we represent that STR as a cycle, we are not directly recording the repeat count. So we are already hiding identifying information just by using a graph representation. But this doesn't work for other cases, like if there's a rare insertion of sequence in your genome, and there's just this alternate path in the graph, this is highly revealing, you just see it. So one way to deal with this is by obscuring that variant by adding strategically designed noise edges and vertices to the graph. Then that adds alternate walks in the vicinity of the variant. You might not be able to directly observe it. You can imagine formulating this problem more rigorously, perhaps to obtain privacy guarantees. When you come in with a read, okay, you align that read to the graph to unlock the variant if that read contains the variant. So you only really recognize the variation if you have a read that contains the variant. So now you should have some insight into how graphs, the utility of graphs for representing genome variation and potentially as a tool for protecting genomic data privacy. But how do you align reads to graphs? And that's the topic I'll discuss next. A problem of string matching and label directed graphs. And this is a very general problem. You can imagine it accompanying, such an index accompanying a graph database. And it's how can we index any finite digraph G whose edges are labeled on some alphabet omega for one, answering the decision problem of whether there's a walk matching a query string Q in G at time linear in the length of Q, independent of the number of edges in the graph and the number of vertices in the graph, and subsequently, two, locating the terminal vertex of each matching walk efficiently in time O of log V with reduced space. So got a query string, we want to know if it's in the graph, we want to know where it is in the graph, okay? Now, if this problem is so important in genomics, you might then expect, well, hasn't it been solved already? And indeed, there is software VG by Yoni Siren, Eric Garrison, and others that aligns reads to graphs, but I would say it solves this problem incompletely because it indexes graphs like they're strings, okay? And what it does is it enumerates all walks in these graphs up to some maximum length, and then it indexes them with a suffix array, or compressed suffix array, and then it layers on graph structure with auxiliary data structures that encode it. Okay, but I want to index a graph like it's a graph, arbitrary graph, an arbitrary alphabet, and importantly, to accommodate arbitrary query lengths. So here I have an example of a graph on the left, and then I have some query examples on the right, along with the terminal vertices and matches. I've included three perhaps pedestrian queries, C, B, C, C, A, 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 and their terminal vertices. Then I've included a couple of more exotic ones. There's AB, an arbitrary number of times. There will always be a match to AB because of that cycle between vertices 1 and 2 on the left. Also included the digits of pi in binary where A is 0 and B is 1. And while several of the characters at the beginning will match, we'll ultimately find that you don't match the digits of pi exactly in this graph. So let's first attack this decision problem, whether it is a walk matching a query string Q in this graph. And this is solved by the classic Rabin-Scott power cassette construction for converting a non-deterministic finite automaton to a deterministic finite automaton, an NFA to a DFA. But if you don't know anything about automata theory, don't worry, I will break this down for you without it. What I want you to do is imagine standing at all vertices of the graph simultaneously, okay? Now walk across all edges labeled A. You'll land at some subset of vertices. Walk across all edges labeled B, and you'll land at another subset of vertices. These will be the terminal vertices of all walks matching the query string AB, okay? But we can imagine performing all such simultaneous walks across the graph, matching all possible query strings, and representing the result in another graph G prime, whose each vertex represents a different subset of vertices in the original graph G. So these subsets can themselves be represented as bit sets, where a bit is turned on if the vertex is included in the subset, and a bit is 0 if it's not included in the subset. Here, there are 3 vertices in the graph on the left and on the right. That's why there are each of these bit sets accompanying the vertices is of length 3. I'll call these bit sets or subsets or states in this talk. And there's an edge labeled Z extending from subset A to subset B if and only if the subset B of vertices is the set of vertices at which you land when you walk across all edges labeled Z from subset A. So there are 2 to the V possible vertices in G prime because there are 2 to the V possible subsets of the vertices of G. We can represent G prime, this transformed graph, as a matrix where columns correspond to different edge labels and rows correspond to different vertices of G prime themselves, potentially labeled by these states, these bit sets. The elements correspond to edges, and they can be represented as pointers from row to row in this matrix. We can do these sample queries just looking at the graph G prime on the right. So let's look for BCC. We start by standing at all vertices of graph G at the same time. That means starting at the all ones bit set in G prime. You see my mouse? Yeah. We walk across edge B. get to this 110 bit set. And then we try to walk across an edge labeled C, but there is no edge labeled C. So BCC is not in this graph G, we conclude. Now let's look for BBA. So we again start at this 111 bit set. We walk across B, we walk across B, we walk across A. It is indeed present. Good. And if we store these bit sets along with all the rows here in this matrix, we can recover the location of the locations of matches. Here, there's a match at vertex 1. I'm just reading it off this bit set here, 0, 1, 0. 0 is the 0th bit. 1 corresponds to vertex 1. Second bit is vertex 2. But if we're doing this, if we're trying to index genome, like pan-genome graphs, these are going to be massive graphs. They're going to have billions of vertices. So this is kind of impractical because we'll be storing a distinct, like a different billion, like size billion bit set for each of these rows, each vertex of G prime. So how can we avoid storing these bit sets of G prime and still recover the terminal vertices of matches? And so this speaks to that second problem that I wrote, locating the terminal vertex of each matching walk efficiently with reduced space. And the solution that Reed, Austin, and I came up with is as follows. You start with some graph G. In this example, the edges are labeled on the alphabet omega equals A, B, C. Now I want you to augment G with 8 edges labeled on some sentinel alphabet omega S that shares no characters with omega, such that A, the edges form a cycle including every vertex in V, and B, that cycle matches a necklace, a circular string where each character has a predecessor and a successor, but no character begins or ends the string. That circular string should be aperiodic. So, and this will mean that each vertex has an identifying tag along the cycle. So let's break this down. Sentinel alphabet. What we're doing is augmenting G with edges labeled on a sentinel alphabet. In this example, that sentinel alphabet is $#, has no characters in common with the alphabet of the original graph, which is A, B, and C. Now, we're augmenting G with a cycle. That cycle will include all the vertices in the graph. And that cycle will match a circular string that's aperiodic. So a periodic string, that would be $#, for example. Here, we're saying it's $$#. What we're doing is assigning a distinct identity to each vertex of the graph using a tag along the cycle. So for vertex 0, you'll have an identifying tag as hash. You walk away along this, what I'm calling identifying cycle, walk away from it, you get that hash. It's the only vertex in which you just like walk away on a hash labeled edge. Then for vertex 2, it would be $#, and for vertex 1, it would be $$. So each vertex has a distinct tag along this identifying cycle. Now, do the power set construction just like before to obtain what we call the nth G prime of G. You can prune components that don't aid in string matching, but I don't want you to worry about that. Instead, what I want you to do is understand conceptually that this transform, this nth, is invertible. Because the vertices of G are specified by those tags along the identifying cycle, and you can discern states from these tags. The tags specify state bits, these bit sets of G prime. And I'll walk you through how to recover this graph G from G prime in the following slides. So let's first obtain the number of vertices in G from this transformed graph, this nth G prime. To do this, we start at some arbitrary vertex, form an arbitrary walk on edges labeled on the sentinel alphabet. And then we do that until we've walked 2 periods. Because what that walk will always correspond to is a set of simultaneous walks confined to the identifying cycle in G, okay? So let's just perform this walk. I walk across hash, I walk across $, I walk across $, again. Then I walk across hash, I walk across $, I walk across $, again. Whoop, 2 periods, my walk length is 6. I divide by 2, I get the number of vertices in G, which is 3. I can also obtain a state bit set from G, okay? And what I do here is I just perform all walks that are on this sentinel alphabet away from a given vertex and pick out these identifying tags from that walk. So here, if I'm trying to identify the bit set associated with the bit I'm pointing, or the vertex I'm pointing at, well, I walk away one way and I get hash that I'll find as an identifying tag. I can walk away another way, I'll get $, and I'll walk away a different way, I'll get $hash. That's 3 distinct identifying tags. And I know that there are 3 vertices in my original graph. So I conclude that this green vertex is the all ones bit set. But now that I have concluded this, I can also walk away on sentinel alphabet labeled edges from any other vertex, perform these locating walks and pick out these identifying tags, walking away from that vertex and thereby infer their state bit sets. Then confining my attention to those bit sets that include exactly 1 bit that's turned on, okay? I can look at the edges extending away from those vertices, and from that infer all the edges and vertices of the original graph. So we're able to perform these locate queries. We can infer the state bit sets from this graph. But I told you that this was a compressed index, okay? Where is compression coming from? The idea here is that there are going to be vertices without edges on the sentinel alphabet that point at the same vertex. And what that's saying is that we bit sets, we share the same subset. So you have this transformed graph where you have a bunch of these vertices and they're all pointing at each other on the sentinel alphabet. And they're all saying, hey, we share subsets, we're sharing subsets. And we're representing subsets as shared via other subsets. And that's where the compression in this index is coming from. But as I said, that number of vertices in the original graph is massive. It's going to be in the billions. How are we going to choose this identifying cycle right? Because if I chose the naive extrapolation of what I wrote $hash to an arbitrary number of vertices like $, hash at the end, that's not going to be so great, because to identify any one vertex, I'll have to walk all the way to my hash. And I could be really far away from that hash when I'm performing a locating walk. On average, it'll take half V steps, where V is the number of vertices in G. So what we want to do is minimize the expected tag length along the identifying cycle, so that identifying any given vertex of G from G prime takes an economy of steps. And that brings me to the last part of my talk on random-like sequence generation. Now, for the remainder of this talk, when I use the word sequence, I'll mean a circular string on some alphabet size k. And that alphabet will always be an alphabet of integers between 0 and k minus 1 inclusive. Sometimes just call it the alphabet k, but I'll always refer to it as k enclosed in these brackets. When I use the word substring, I'll mean a string of contiguous characters in a sequence. An example sequence is on the left, it's of length 8, and all 8 of its length 3 substrings are on the right. Talking about circular strings here. All right, so consider drawing each character of a length L sequence uniformly at random from this alphabet K. Now, it's equally likely for any length M substring to be at any given position, and there are k to the m possible strings of length M. So L over k to the m is the expected number of occurrences of any length M string as a substring. Now, it turns out that there are random-like sequences that satisfy this criterion of L over k to the m being the expected number of occurrences exactly. And the canonical example is a De Bruijn sequence of order n on the alphabet k. Its length L is k to the n. And it contains every possible length n string exactly once as a substring. So it's optimally short because there are k to the n possible such strings of length n, and the sequence is of length k to the n. That makes it ideal for this identifying cycle to add to the graph G, because that tag length is going to be the order of the De Bruijn sequence, N, which is log base K of L, the length of the sequence. This only works when that length is a power of K. But we'll have an arbitrary number of vertices in our graph. So how do we generalize De Bruijn sequences to arbitrary lengths? That's what this last part of the talk is about. But let's focus on De Bruijn sequences for a little bit longer. Turns out that sequence I showed you a few slides ago is an order 3 binary De Bruijn sequence, 0, 0, 0, 1, 0, 1, 1, 1. Contains all possible length 3 strings on a binary alphabet of substrings, and they're displayed in their order of appearance on the right. So taking a closer look at De Bruijn sequences, if every possible length n string occurs exactly once as a substring, then every length n minus 1 string occurs k equals k to the n over k to the n minus 1 times as a substring. Every length n minus 2 string occurs k squared equals k to the n over k minus 2 times as a substring. So on, all the way down to how every length 1 string occurs k to the n minus 1 equals k to the n over k times as a substring. You can see this in that order 3 binary example here. There are 2 instances of each length 2 string on the alphabet 2. You can see 2 instances of 0, 0 here on the left. There are 4 instances of each length 1 string on the alphabet, the binary alphabet. So they're like 4 zeros and 4 ones. So that's telling us what's happening in an order n De Bruijn sequence for substring sizes. What's going on here? Oh yeah, for substring sizes smaller than n. But what about substring sizes that are larger than n? And so if m is greater than n, then k to the n over k to the m, the expected number of occurrences, would be between 0 and 1. It's not an integer. And what happens is there's either 1 or 0 instances of any such string. And include the length 4 example here, you're either in or you're out. Half of all possible length 4 strings occur as substrings of this De Bruijn sequence, and half do not. Oh, okay. So that leads us to this alternate definition of De Bruijn sequences. So we can look at an order n De Bruijn sequence on this alphabet K as a sequence of length k to the n, such that any string on k of length m less than or equal to k to the n appears either k to the n over k to the m times rounded down to the nearest integer, or k to the n over k to the m times rounded up to the nearest integer as a substring. And it turns out this alternate definition of De Bruijn sequences leads to a very natural generalization to arbitrary lengths. So Rachel and I defined a PKL sequence on K to be a sequence of length L such that any string on that alphabet of K of length m less than or equal to L appears either L over K to the m times floored, rounded down to the nearest integer, or L over K to the m times sealed, rounded up to the nearest integer as a substring, okay? So, it's all well and good to define a sequence, but that doesn't mean that that sequence exists. Now we do know that De Bruijn sequences exist for all possible combinations of the order n in the alphabet size k. And this was proved first by explicit construction by Monroe-Martin in 1934. So do PKL sequences exist for all possible combinations of the length L in the alphabet size k? And the answer to this question is yes. And this was proved by Rachel and me. We obtained an O of L time, O of L log K space general algorithm for its construction. And the central tool of this algorithm is Lempel's lift, perhaps known more widely in the literature as the inverse of Lempel's dimorphism. Yes, this is the Lempel of Lempel-Ziv, who unfortunately passed away earlier this year. The gist of this is, this Lempel's lift is, you take an integral of a sequence modulo k. When I use the word integral, I really mean cumulative sum. And when I use the word modulo k, what I mean is you do your arithmetic and then you take the remainder from dividing by k. So given a sequence S of I on the alphabet k, write this integral R of L equals some integration constant, which is going to be some number between 0 and k minus 1 inclusive. Plus this cumulative sum. You're just adding a character, adding another character in that cumulative sum, getting a new sequence. So what this does is it affects a 1 to k map from the set of length m strings into the set of length m plus 1 strings for every m. And we can make bigger de Bruijn sequences by integrating smaller de Bruijn sequences modulo k. And might be confusing on this slide, but it's best learned by example. So let's build de Bruijn sequences with Lempel's lift. We start with an order 0 de Bruijn sequence, okay? It's 1. Integrate 1. How do you integrate 1? Well, you choose an integration constant, we'll choose 0. And then you just start going around the cycle and adding 1. So it's 0, I add 1, I get 1, I add 1 again, I get 0, I add 1 again, I get 1. I'll keep getting 0, 1, 0, 1, 0, 1. One period of this is 0, 1. That is an order 1 de Bruijn sequence, one instance of 0, one instance of 1. Now let's integrate 0, 1. Start with an integration constant 0 again. I'll add 1, I'll just start at this 1, and I'll get 1. Add 0, I'll get 1. Add 1, I'll get 0. Add 0, I'll get 0. Add 1, I'll get 1, and so on. I'll just keep getting 0, 1, 1, 0. One period of that is 0, 1, 1, 0. And if I started with the integration constant 1, instead, I get the same thing. That's an order 2 de Bruijn sequence. Now, integrate 0, 1, 1, 0. Well, if I start with the integration constant 0, I'll get the result 0, 1, 0, 0. But if I start with the integration constant 1, I'll get 1, 0, 1, 1, its complement. Now this is 2 cycles. This isn't a de Bruijn sequence, so what are we supposed to do now? And the answer is this first trick, which is cycle joining. What we're doing is we're taking 2 cycles and we're literally joining them at a shared substring. We can affect this by concatenating rotations of the cycles whose prefixes are the same length n minus 1 strings. Here, our target order is 3, length n minus 1 is 2. And it turns out that the natural choice for the shared substring at which you're supposed to join these cycles is an alternation between zeros and ones. So here, we have these 2 cycles, 0, 1, 0, 0, and 1, 0, 1, 1. We rotate the length 2 alternation to 0, 1, for example. And we get 0, 1, 0, 0, and 0, 1, 1, 1. Concatenate, we get 0, 1, 0, 0, 0, 1, 1, 1. That is an order 3 de Bruijn sequence. Yeah.
Please go back to the previous slide.
For sure.
Do you mean the 0, 1, 0, 0 is order 2 de Bruijn cycle? The one which is pointing at. Yeah, the 0, 1, 0, 0.
So 0, 1, 1, 0 or 0, 1, 0, 0?
0, 1, 0, 0.
So 0, 1, 0, 0 is one of the possible integrals of 0, 1, 1, 0. That is not a de Bruijn sequence. So what we have are 2 sequences that aren't de Bruijn sequences, but that collectively have the right substring content to make a de Bruijn sequence. So when we join them, we can form a de Bruijn sequence if we join them in some shared length n minus 1 substring. So that's what I'm doing. I'm trying to take these 2 cycles that have the right substring content of a de Bruijn sequence and make them into one cycle while preserving that substring content. That is the effect of joining.
Thank you.
Sure. Good question. So now let's do this integral again. I'll start with that order 3 de Bruijn sequence, 0, 1, 0, 0, 0, 1, 1, 1. Integrate it, and I'll get 2 cycles again, like before. But now the length of the substring I want to join on is going to be 3, 1 order up, not 2. So we'll rotate to that alternating prefix. We'll choose that prefix to be 1, 0, 1. We rotate in both cycles. We'll get 1, 0, 1, 0, 0, 1, 1, 1, 1, 0, 1, 1, 0, 0, 0, 0. Join those 2 cycles, concatenate those 2 rotations, and we'll get an order 4 de Bruijn sequence. So in the general binary case,
how do you choose the integrate?
What's that?
How do you choose the integrate?
Okay, so you're talking about the integration constant? Yeah. So the whole thing is that here, when I'm doing this integral, I'm choosing both. I'm doing all possible integrals. If I choose the integration constant 0, I get the top guy. If I choose the integration constant 1, I get the bottom guy. But there are actually 2 possibilities. So when a cycle has an odd number of ones, that integral is unique and it's self-dual, meaning that it equals its complement. So if I chose either 1 or 0, I just get the same cycle. But when the cycle has an even number of ones, there are 2 integrals and they're complementary. But the total length always doubles the cycle length. The total length of the integrated sequence spans exactly double the characters as the sequence you're integrating. All right, so that's how we can construct de Bruijn sequences using length pulse lift. But we need one more trick to be able to construct these PKL sequences we've defined. And I'll go over how to do this for PKL sequences for k equals 2 on a binary alphabet. The trick is we insert 1 in the longest string of ones as necessary after integrating. And when I say as necessary, I mean that there is a prescription for doing this obtained from the binary representation of the length of the sequence L. Again, best learned by example. So let's try to generate a length 13 sequence, a length 13 PKL sequence for k equals 2. In binary, 13 is 1101. I want you to just ignore that leading 1. Binary, you'll always have a leading 1. Start as you would for de Bruijn sequences. Integrate 1. You'll get 0, 1. Now this pink 1 is telling you to insert 1 in the longest string of ones. So we do that, 0, 1, 1. And then we perform another integral. We'll get 2 cycles, 2 possible cycles, depending on the integration constant we use. 0, 1, 0, and 1, 0, 1. Rotate and join at the substring 0, 1. And we get 0, 1, 1, 0, 1, 0. Now this green 0 is telling us to insert nothing. So we do that, we insert nothing. We integrate 0, 1, 1, 0, 1, 0, and get this length 12 sequence. And the final one here in the binary representation of L is telling us to insert 1 in the longest string of ones again. And that longest string of ones is 1, 1, 1. We insert a 1 there, we get 1, 1, 1, 1. And now this is a length 13 sequence. It's a P2L sequence. And you can verify it is, because it should have 13 over 2 to the m floor instances of every length m binary string, and 13 over 2 to the m sealed instances of every length binary string, or one or the other. So there are 6 or 7 length m strings, length 1 strings occurring as substrings, like 6 zeros, 7 ones. They're either 3 or 4 of the length 2s, they're either 1 or 2 of the length 3s, and they're either 1 or 0 of the length 4s. So this checks out. This is a PKL sequence. So you understand operationally how this is going. Probably with these slides, you could recover this. You could construct PKL sequences of your own or P2L sequences, but you might not understand why this. The keys to an inductive proof are that both integration and 1 insertion preserve P2L-ness. Integration works because you get this 1 to 2 map. Each length m string occurrence gets mapped to 2 length m plus 1 string occurrences, while the total length of the cycles is multiplied by 2. And 1 insertion preserves P2L-ness because your substring content is basically the same, except you're going from 0 instances of an all 1 string to 1 instance of that all 1 string. And you're permitted both either 0 or 1 instance. So there are actually details I've kind of glossed over even in the binary case, but if you're interested to know how this generalizes to PKL sequences, maybe it's a puzzle you want to solve on your own, but you can also visit bit.ly/PKLseqs, where we have Python for generating the sequences. We include a link to our paper. You can also visit debruinsequence.org, which is Joe Sawada's site. Joe is an expert in combinatorial generation at the University of Guelph, and he took a liking to this work and ported our Python to C. And also, he also offers clear explanations of this universe of random-like sequences, including de Bruijn sequences. So it's definitely worth checking out. We've arrived at kind of a conclusion, a complete solution, this compressed graph index. So how can we tell whether a query string is in the graph and where that query string is in the graph? And our solution is, number 1, we add an identifying cycle to the graph that matches now a PKL sequence on some sentinel alphabet K. For L, the length of that pKL sequence is equal to the number of vertices in the graph. And we do this power set construction as usual to form the nth G prime. And now we can form string matching in time linear in Q, size of the query string, but also locate queries, basically identifying the terminal vertices of matching walks in the graph, in O of log V time per vertex, because we're using a pKL that identifying tag is going to be of length log V. So there's a lot of directions in which this work can be taken. On the graph search side, on an implementation of the index, but it would greatly benefit from more compression strategies. This could include just sort of ordering and compressing rows of the matrix representing the nth, but we can also think about, for example, in the original graph, we might want to merge different paths. Graphs themselves provide a means for compression, essentially the problem of NFA minimization. I told you how to do exact matching, but not how to do approximate matching. And there are going to be variants in these reads that were aligned to graphs that aren't present in the graph. So it's not only solved by this exact matching problem. It's going to be good strategies for like pulling down subgraphs if you want to do approximate matching. The sequence side, there are theoretical questions and applied questions that are interesting. Like no one has counted, because I guess we introduced them, the number of pKL sequences there are for fixed alphabet size K and L. These have been counted for De Bruijn sequences, famously by De Bruijn and Tatiana van Aarde and Ehrenfest. We could also try to use less space for generation. I provided an O of L log K in space algorithm, but De Bruijn sequences can be generated in constant space per additional character. So there's a lot of room between these two extremes, and you might think of an entirely different framework for constructing pKL sequences. Now there's a class of problems that might be of interest to folks at Galois in genomic data privacy. This is like an eminently fundable space because it's in our national security interest to protect genomic data privacy. So there's that variant obfuscation problem that I talked about, but one class of problems I didn't talk about is homomorphic encryption for genomic data analysis. You can imagine, having submitting your encrypted genome to a server. That server does some useful computation over that while it's encrypted, and then gives you some summary results that are useful. And some problems that folks have worked on in the space are genotype imputation, where they're inferring variants that weren't submitted to the server, and disease detection. So this is a wide open space. If you're interested in any of these problems or technical problems that are related to these, or you're just excited about your work and think I might be interested too, please just write me. That's my email address. And I'd like to acknowledge my collaborators, Austin Wynn, Reed Thompson on the graph search problem, Rachel Ward in sequence construction, Institution Oregon Health and Science University, and the University of Texas at Austin for awarding me an Odin Faculty Fellowship, allowing me to just sort of spend some time there, supported some of the research contained here. So great, I'll take questions. Okay, I guess I might have been long. I apologize for that.
Hey, I've got a question. So you told us to avoid storing 1 billion bits per state. But it seems like you still have the problem of having 2 to the 1 billion states. So what do you do about that?
Excellent point. So you would look at, maybe I had like a slide on this. So you'd look at, for instance, like this matrix here, and I'm storing a matrix that includes these extra sentinel alphabet vertices. If there's 2 to the V states, indeed, you're not saving space at all. But if you ever 2 to the V states, you wouldn't even be storing the bit sets because you could just store them in order according to 1, 0, 1, 2, 3, 4, 5, if there were 2 to the V states. So you have to be operating in a regime that's between 2 to the V vertices and something that scales more linearly. So if, for instance, if you're working with a suffix tri, the number of vertices in the suffix tri scales as n squared in the size of a string. So even naively, you get something in string-like version, string-like parts of a graph. You'd be indexing them and you wouldn't be seeing this exponential blow up. And you can even, like you can represent it as a directed acyclic word graph. So you're going to be in a regime where you scale linearly with the number of vertices, you scale exponentially, and that's a pretty wide range. The way these graphs are going to work is there are a lot of like string and tree-like structures in the graph, but then there are going to be these hairballs where you are blowing up exponentially. But this is a useful tool when you're operating in a useful regime that's between the worst case and the best case for strings. It's a great question though. It's like the first thing, it might be the first thing you think of, and I kind of just glossed over it.
Thanks.
Any more?
I was thinking, so in my university, like I remember we generated the De Bruijn sequences using finding like Eulerian cycle in a graph. So how does the complexity of that generation varies with whatever you have?
So, so linear time is the best you can do. So when you're doing, I think you might be doing like there's like a DFS, you're just doing a DFS, that's not going to scale as favorably at all. But there are lots of ways to generate De Bruijn sequences. For instance, you can, there are these concatenation constructions where you're concatenating all Linden words whose lengths divide the order, the sequence in lexicographic order. And those concatenation constructions are super efficient. There are also shift rules. Those are the ones that give you those O of 1 space per bit generation. You'll find like a web of different De Bruijn sequence constructions on that debruijnsequence.org website. There's some people who take De Bruijn sequences very seriously. And they are beautiful sequences. Okay, great. I thank you all for coming. Reach out if you're interested. This is my email address.